This third section of evolution notes will be on the origin of cells. Scientists have been trying to investigate the origin of life for quite a long time now. One of the early theories was called the theory of spontaneous generation. This meant that life could just arise from nothing, right? It could just spontaneously generate. And so there have been several experiments from the past that have tried to investigate this concept. Is spontaneous generation of life possible? I will play a video that will go through two experiments. The first will be on Francisco Redi's experiment. The second experiment will be on Pasteur's.
Where do cells come from? If a cut of meat is left out, over time it will putrefy and begin to teem with microorganisms and possibly with larger organisms like maggots. Prior to the mid to late 19th century, the origin of microorganisms in decaying matter was in question. Some maintained that microbes arose from other microbes that landed on the food from the air. Others supported the hypothesis of spontaneous generation, which states that living organisms can arrive spontaneously from non-living matter. In the 1600s, Francisco Redi sought to test the hypothesis of spontaneous generation by applying what came to be known as the scientific method, a process of making observations, asking questions, formulating a hypothesis, and designing experiments to test the hypotheses. Redi and others observed that flies and then maggots could be seen around pieces of meat that were left out in the open. He therefore asked the following questions. Where do the flies come from? Is the rotting meat transformed into the flies? From these questions, Redi formulated the hypothesis that only flies can make flies, and that rotting meat cannot be transformed into flies. Redi sought to test his hypothesis by performing the following experiment. He placed pieces of meat in 3 glass jars. The first jar was left open, the second was covered with a loose netting, and the third was completely sealed. All jars were exposed to flies in the surrounding room. Redi predicted that if meat could not be transformed into flies, then the sealed containers should not produce either maggots or flies. Whereas, if the meat can be so transformed, then the sealed jar should also develop maggots and flies. Redi recorded the presence or absence of flies and maggots in each of the 3 types of jars. As he predicted, neither flies nor maggots were found in the sealed jars, whereas in the open jars, maggots and flies were abundant. In the jars covered with netting, maggots were found within the netting itself, but not on the meat inside the jar. Redi concluded that meat could not transform into flies. Only flies could produce flies. The theory of spontaneous generation could not be supported and was incorrect. Unfortunately, Redi's experiment did not convince everyone. Some argued that while spontaneous generation might not apply to larger organisms like maggots and flies, it might still be applicable to smaller microbes. The question was finally answered definitively in the late 1800s by Louis Pasteur in his now classic experiment. Pasteur's hypothesis was that if cells could arise from non-living substances, then they should appear spontaneously in sterile broth. To test his hypothesis, he created 2 treatment groups: a broth that was exposed to a source of microbial cells and a broth that was not. For his control treatment, Pasteur used a straight-necked flask that allowed particles in the air to fall into the broth stored in the flask. For his experimental treatment, Pasteur used a swan-necked flask. The neck shape and length assured that no cells could enter the broth from the air. Pasteur then performed the same procedure on these 2 flasks. He boiled the broth to kill any existing organisms. He then let the broth cool and allowed it to sit for several days. after which he removed the necks of the flasks and checked the broth for the growth of any organisms. Pasteur found living organisms only in the control flask. Because the experimental flask remained sterile, the hypothesis of spontaneous generation was rejected. By changing a single variable, the shape of the flask, Pasteur was able to conclude that cells were not generated spontaneously, but were actually entering the broth from the surrounding air. Microorganisms, carried by dust particles, fell into the straight-necked flask. However, the swan neck trapped the particles, preventing cells from entering the broth.
So again, in Pasteur's experiment, he wanted to test whether or not microbial growth would occur in sterile broth. So he designed an experiment with the two swan neck flasks. On one of the swan necks, he broke it so that it was a straight neck. And then he heated up the broth in each of the flasks to sterilize it. That way he could be sure that there were no microorganisms in it to begin with. Then he let it sit for a period of time. When he came back, one of the solutions was cloudy. That was the broken neck flask. And the other solution was still clear, meaning that there was no microbial growth in this flask, but there was microbial growth in the cloudy flask. Cloudy is an indication that microbes are there. He concluded that you did need a source of contamination, which was the dust, to bring in the microorganisms to be able to have them grow within the flask. This was only possible in the broken neck flask because the dust could just fall in with the microorganisms. In the swan neck, the dust accumulated down here and therefore could not travel up and make it into the broth. So therefore, he did ultimately disprove spontaneous generation of microorganisms. But does this mean that spontaneous generation of life could never occur? Well, what about the first cell? Remember that the cell theory we learned about in BioH has three parts. Number one, all living things are composed of cells. If they're not made up of cells, they're not considered alive. Number two, the cell is the smallest and most basic unit of life. Nothing below the cellular level can fully function on its own. And number three, cells arise from other cells. Cells divide to make more cells, and that's how we get more cells. One thing we know about cells is that cells can only be formed from the division of pre-existing cells. We know this to be true. We've seen this in lab. We know cells multiply through cell division. We've learned about two different types of cell division. Mitosis results in genetically identical diploid daughter cells, whereas meiosis generates genetically different haploid daughter cells. Meiosis makes the gametes, which are used in sexual reproduction. Either way, cells come from the division of pre-existing cells. So other than Pasteur's experiments and experiments that tested spontaneous generation prior to Pasteur, what other evidence do we have that supports the cell theory? One, cells are incredibly complex structures. In fact, in lab, we have not been able to figure out a mechanism where we're able to make cells from simpler subunits. The only known mechanism for cells being made is when other cells are already present and then they divide to make more cells. Number two, all known examples of growth, whether we're talking about growth on a tissue level, which is when cells divide to make more cells of the same kind of cell, or as an organism is growing, for example, you are made up of more cells today than you were 5 years ago because you have grown through cell division, or as a population, right? If we think of how many organisms are out there as a population reproduces, all of that is still just due to cell division, right? So all known examples of growth, whether we're talking about on a small level or a much larger level, they're all due to cell division. Number 3, viruses are not made up of cells, therefore they are not considered to be living organisms. Viruses can be made up from simpler subunits. However, they cannot do this without using a living organism to assemble those parts together into new viruses. A virus has to invade a host cell, invade a living cell. And then once it gets its parts inside of that cell that it has infected, then it can assemble more viruses. So again, viruses are not considered alive because they cannot divide to make more viruses without the help of a host cell. Number 4, the genetic code is universal. There's only 4 nucleotide bases in DNA, 4 in RNA. Some of those overlap. This leads to the 61 codons that code for amino acids. The other 3 codons, remember, code for stop codons. And so we share this genetic code. It's universal between all living organisms. Remember in BioH, we were able to take DNA from a sea anemone, insert it into a bacterial chromosome, and that bacteria could make the proteins that belong to a sea anemone. Again, because there's a universal genetic code. The logical deduction is that all cells must have came from a common ancestor way back in time. If we accept the fact that there must have been times in history when there wasn't cells, when there were no living organisms, then we must accept the fact that the first cells likely came from non-living material. The only other possible explanation to the first cell is that life got here from somewhere else in the universe, and then once it got here, divided through cell division to make more. So it has been really difficult for us to figure out what actually happened here on Earth billions of years ago when the first cell actually formed. There are key problems when we try to figure this situation out. Some of the key problems are listed below. Number 1, how do we get simple organic monomers to form from non-living inorganic elements or atoms or molecules? That has presented challenges. Number 2, how do we get those monomers of organic compounds to then assemble into stable polymers like DNA, RNA, complex carbohydrates, phospholipid bilayers, protein chains? How do we get that to occur? Number 3, how do we get those polymers to also make more polymers? They have to be able to self-replicate. Replication is the basis of inheritance. Every time DNA replicates, it gets copied first, right? And then gets passed on to the next generation. Well, how do we get these polymers to make more of themselves? And then number 4, how do we get the membrane, the phospholipid bilayer, to package around all of those substances to allow for this process to continue to occur? Earth's early atmosphere was very different than it is today. Earth's early atmosphere was considered reducing in the early stages. It did not contain any oxygen gas, and it did not contain any ozone. None of these gases started to really develop in high concentrations until plants started photosynthesizing. And once we had plants, that's when the concentrations of these particular molecules increased greatly, allowing for different niches that are more similar to what we have going on today. So here are some of the molecules that were present in Earth's early atmosphere. Remember, none of them are O2. Can you identify what these molecules are? I'd pause the video at this time and think about it. So hopefully you came up with some of these: hydrogen gas, N2, water vapor, methane, which is CH4, ammonia, NH3, CO2, carbon dioxide, and then hydrogen sulfide. These gases likely came from a lot of volcanic activity that is thought to have happened in Earth's early days. Of all of those gases, the most abundant gases were CO2, methane, and water vapor. All of these gases are considered to be greenhouse gases, which means they have the ability to trap heat and keep heat overall on Earth. Coupled with this, there was no ozone layer. Our ozone layer today acts as a filter for some of the UV radiation that is coming in from the sun. Without that filter and having such high concentrations of potent greenhouse gases, it did allow for a very high temperature to occur in early Earth. And so there had to be somewhere on Earth where this was happening that essentially allowed for the first cell to form. During this time, there was also thought to be a lot of storms, so a lot of lightning, that electrical energy that was present, and that is something else to consider as well. Here are just pictures of showing how different Earth's prebiotic, before life, atmosphere was compared to today's atmosphere. At some point, monomers would have formed, and these monomers must have formed from chemicals that reacted together to create the monomers and ultimately led to polymerization that led to the biological molecules that we know are essential to life today. So carbs, proteins, nucleic acids, and lipids. This has been difficult to kind of figure out how this could have happened, but there have been experiments that tried to recreate the conditions that would allow for this synthesis several times throughout history. Miller and Urey is one group of scientists who did try to create these conditions in lab in 1953. They were trying to figure out if this chemical evolution, all these inorganic chemicals, could come together to create more complex chemicals that were essential to life. So they took all the gases that were present in Earth's early atmosphere and put it in a closed vessel container, which simulated the early atmosphere. They heated it up with water. This heat was like the volcanic activity that would have been present during that time. And then they also allowed electricity to enter and spark these gases. And that was supposed to be like the lightning. So the condition that they didn't really mimic was like the intense bombardment of UV rays, but all the other things they did mimic, and they tried to see, could they potentially get monomers to form? After a week, they found that they did. 13 of the 20 amino acids did actually form. About 15% of the carbon that used to be inorganic was now in an organic form. So that was pretty big. And of those organic compounds, about 2% of them were the amino acids. That means there were other molecules that formed as well, other organic molecules, like some simple lipids, for example, formed as well in the solution. So next we'll watch a video explaining the experiment of Miller and Urey.
What's up?
Hi, Jira. We brought you here today.
In the 1920s, Oparin and Haldane hypothesized that the atmosphere of the primitive Earth contained gases such as nitrogen, ammonia, methane, carbon dioxide, water vapor, and hydrogen. Oxygen did not become a part of the atmosphere until the advent of photosynthesis millions of years later. It was further hypothesized that under these conditions, organic molecules could be formed if there were a source of energy such as electricity from storms or ultraviolet radiation from the sun. In 1953, Stanley Miller and Harold Urey tested this hypothesis. They constructed a closed system with a reservoir for water, sampling ports, a chamber for the input of electrical energy, and a condenser to convert the water vapor back to a liquid. The researchers then added water, hydrogen gas, methane, carbon dioxide, ammonia, and nitrogen. The water was heated to form a vapor, and electrical sparks were generated to mimic lightning. This apparatus simulated the possible conditions of the early Earth. When the water was analyzed after 1 week, they found simple carbon compounds such as formaldehyde and hydrogen cyanide. These compounds then combined to produce formic acid, urea, and amino acids. In similar experiments carried out over the next several years, various organic compounds have been synthesized. These experiments showed that simple organic compounds, including the building blocks of proteins and other macromolecules, could be formed from gases present in the primitive atmosphere with the input of electrical energy.
So here's what their experiment looked like. They created an experiment under low oxygen conditions to simulate the reducing atmosphere, and they inputted a whole bunch of gases that were thought to be present in Earth's early atmosphere. They heated up the experiment, added water, allowed for condensation to occur. They collected the liquid, they added the electrical sparks to show the lightning, and then ultimately, after about a week, they tested the liquid and found that they could in fact have amino acids and some simple lipids join or come together from the inorganic substances that they inputted into the experiment. Therefore, they concluded it was possible that the conditions of pre-biotic Earth could potentially form organic molecules. However, even though they did get some of the monomers to form, as soon as the monomers came together to form polymers, they would immediately hydrolyze. So getting these molecules to assemble into polymers gave future issues. It was hard to get the molecules to do this. So then they were trying to figure out where could this potentially happen. And so currently scientists think that it's possible that deep in the ocean there are these deep sea hydrothermal vents, which are like cracks or fissures in the ocean floor where a lot of heat comes out of and a lot of gases come out of. These are typically found near volcanoes. Because there's so much inorganic molecules that come out of it and it's so intensely hot, it is thought that potentially this could be the place that had the right conditions and chemicals to allow for polymerization to occur, even in Earth's early conditions. In fact, in 1977, they did find living cells near these types of vents. But again, we need polymers that can self-replicate, so make more of themselves. Even though today we know that DNA is the major molecule that provides the information that will be inherited from cell to cell, and it's incredibly stable and good at storing information. One thing we know about DNA is it's complicated to replicate. DNA requires proteins in order to replicate it. DNA requires enzymes to replicate it. So it doesn't really make sense that DNA would be the initial genetic material. So if we consider another molecule, RNA, RNA can both store genetic information and it can self-replicate. RNA can catalyze the synthesis of itself. It can help make copies of itself. It can also help form other types of bonds. For example, in the ribosome, there's a type of RNA called rRNA. rRNA also has catalytic properties. rRNA helps form the peptide bonds between amino acids during translation when a protein is made. So because rRNA acts like an enzyme, which we would call a ribozyme, a ribozyme is a RNA acting like an enzyme. So rRNA would be an example of a ribozyme. Since RNA can do all of these things, it is much more likely that RNA was the first genetic material, not DNA. And so this is going to be supported by a hypothesis that's called the RNA world hypothesis, which we will talk about next. After we had polymers to form, in order to maintain conditions that would allow for self-replication to occur, those polymers would need to be packaged. So they would need to be packaged in a membrane, a phospholipid bilayer that ultimately would create a microenvironment inside the cell that is different from the environment outside the cell. Initially, when these little microenvironments, these little vesicles formed, we call them protocells. A protocell is literally just a membrane, a phospholipid bilayer with water both in and outside of it. Remember, phospholipid bilayers form spontaneously in the presence of water. So if we had phospholipids, they could form this way spontaneously if environmental conditions were right. So aqueous solutions, for example. But this would have to happen, and then it's possible that if these protocells formed, that things from their outer environment, like monomers, polymers, could be incorporated into them. If those things got into them, this could provide the environment that was different enough from the outer environment that led to self-replication and led to more complex monomers and polymers continuing to be built. Note, it is important for you guys to understand that even though scientists do try to investigate these processes that happened early in time, it has been a real struggle being able to successfully test these things in labs. In this case, the exact conditions of prebiotic Earth cannot be replicated. And so on top of this, these initial protocells did not fossilize. So a lot of this is just us trying to investigate and figure out what makes the most sense, what potentially could have happened in these conditions that could have ultimately led to the first cell being formed. This slide shows the RNA world hypothesis. First thing that needs to happen is RNA has to form. So from inorganic substances, RNA forms a polymer. After RNA forms a polymer, it makes more of itself. So RNA can self-replicate via ribozymes, RNAs that act like enzymes. Once we have more copies of RNA, RNA can be used to form proteins. Remember, RNA also acts as a catalyst to form the peptide bonds, like rRNA, for example. Once we have proteins, then these things could be incorporated into the protocell. So now we have proteins, we have rRNA inside of it. This new cell membrane environment creates a chemistry inside that is different from the outer environment. Over time, RNA can encode both proteins and DNA. So from RNA, we make proteins. These proteins, some of them enzymes, can catalyze the synthesis of other reactions. So we could use some of these enzymes to work backwards from RNA's code to get DNA. Over time, long periods of time, DNA ultimately becomes the master template. So again, this is known as the RNA world hypothesis, which states that RNA is likely the first genetic material. Once the first cell formed successfully, then that cell ultimately had to serve as a common ancestor to future cells. The last universal common ancestor, which we call LUCA, is hypothesized to be the common ancestor that leads us back to all the domains we now have today. There are 3 current domains today: domain bacteria, domain archaea, and domain eukaryota. Both bacteria and archaea domains are prokaryotic domains, and eukaryotic domains are more complex. They have a nucleus. More on that in a little bit. So LUCA is considered to be the last universal common ancestor that unites all modern organisms of today. It's suggested that LUCA was very, very simple. Essentially, it was just a phospholipid bilayer with DNA, RNA, and proteins inside of it. Genetic homologies of DNA, RNA, and proteins leads us to evidence that supports LUCA as the last universal common ancestor that unites the modern domain. Later, we'll learn about the theory that supports eukaryotes originating from a combination of bacteria and archaea domains. Although Luca is considered to be the last universal common ancestor uniting the modern species, that does not necessarily mean that Luca was the first cell. There may have been other cells that did form near the time of Luca or prior to the time of Luca, that once Luca did form, it was in competition with those other cells. And those other cells may have been outcompeted due to natural selection and other processes, and therefore they went extinct when they had to compete with Luca. So you can see here that there are other branches and other organisms prior to Luca, likely, but that Luca is the last universal common ancestor that unites the modern species. Evidence supports Luca as not only the last universal common ancestor, but also that Luca likely formed in those deep-sea hydrothermal vents. 355 different protein families have been identified from bacteria and archaea to have been shared with Luca, so homologies. These genes would have been important genes. These genes would have been genes that Luca would have needed for anaerobic metabolism, since at that time the atmosphere was low in oxygen, and for fixing carbon dioxide and nitrogen so that they could ultimately make organic compounds. From this information, it is deduced that Luca probably lived in an environment that was high in the concentration of Earth's early gases, and that would have had the need to be able to make simple molecules from inorganic compounds. This places Luca, again, in those deep-sea hydrothermal vents where conditions are thought to have been ideal for the first cell formation. And this is thought to have happened over 3 billion years ago. Most scientists agree that Earth formed about 4.5 billion years ago, but trying to figure out when life formed after that has been difficult to figure out. There are isotopes that scientists can use to help date carbon molecules that have been fossilized in rock in order to figure out how old were these earliest cells. Typically, they use radioactive isotopes of carbon or other types of isotopes, ratios of different types of isotopes, to provide an estimated age of these particular early cells. In Australia, we have discovered mats of cyanobacteria that formed stromatolites. Stromatolites are essentially almost rock-like structures that are actually made up of living organisms. And these also happened in the oceans, in shallow oceans. Cyanobacteria are blue-green bacteria. Because we have dated these cells, these early cyanobacteria cells, to have been formed about 3.42 billion years ago, this would provide the minimum date for the earliest cells, and therefore a minimum date of the age for Luca. Remember, there are likely to have been cells that have formed prior to Luca that were much more simple, and so the earlier, more simple cells would be even older than this. Scientists also use genomic information to try and date the origin of cells. So this is when they're going to use the number of similarities or differences between DNA between the different species. And the amount of differences between the genomes of two different species is proportional to the time that they diverge from a common ancestor, meaning the more differences that has occurred, the more time has passed, and so the older the common ancestor would have been. Using approaches like this, it suggests that the first cells could have formed shortly after Earth formed, so about 4.5 billion years ago. Again, all of this has been difficult to figure out for sure, and scientists are still investigating these processes the best that they can. The origin of eukaryotic cells can be explained by a different theory that's called the endosymbiotic theory. The endosymbiotic theory says that eukaryotic cells likely formed from a relationship, a symbiotic relationship between an archaea and a bacteria. This particular theory would state that a larger archaea organism likely took in a smaller bacterial organism and let it live inside of it. So instead of breaking it down and killing it, eating it for nutrients, it let it live inside of it. This is because this particular cell likely provided it a benefit that gave the larger host cell an overall advantage to survival and therefore reproduction. And so when an organism lives inside of another organism for benefit, that's called an endosymbiont. An endosymbiont. This endosymbiont over time developed into organelles. So some of the endosymbionts that were engulfed would have been giving the benefit to the host cell of being able to make a lot of ATP energy. So they were really good at making lots of ATP. They could use oxygen also to make ATP. And so because of this, the host cell would get some of that ATP. And this was also a mutualistic relationship. So the host cell was providing, a place for protection for this organism to live inside of it. And they were truly benefiting each other. Over time, these particular bacteria ultimately evolved into the mitochondria. A similar situation may have occurred where an organism, a smaller organism that was engulfed, was really good at doing photosynthesis. And so it would give sugars, for example, to the archaeoorganism. And so over time, it developed into a chloroplast. Let's watch a couple of videos about these processes. Next, we'll watch this animation over here.
The eukaryotic internal membrane system, called the endoplasmic reticulum, and the nuclear envelope may have evolved from infoldings of the plasma membrane in an ancestral prokaryotic cell. Such infoldings are common in modern prokaryotic cells. The theory of endosymbiosis proposes that a critical stage in the evolution of eukaryotic cells involved endosymbiotic relationships with prokaryotic organisms. Microorganisms that live within other cells and perform specific functions for their host cells are called endosymbionts. According to the theory, energy-producing bacteria may have been engulfed by a larger primitive cell and come to reside within it, eventually evolving into what we now know as mitochondria. Photosynthetic bacteria use photosynthetic pigments embedded in internal membranes to derive energy from sunlight. These bacteria may have come to live within early eukaryotic cells, leading to the evolution of chloroplasts. Several facts provide evidence for the endosymbiotic hypothesis. A few examples include: 1. Mitochondria and chloroplasts contain their own circular DNA, similar to DNA in bacteria. 2. Both mitochondria and chloroplasts are surrounded by two membranes. The inner membrane is probably evolved from the plasma membrane of the engulfed prokaryote, while the outer membrane is probably derived from the cell membrane of the host. 3. Mitochondria are about the same size as bacteria. 4. Mitochondria appear to have been derived from purple bacteria, and chloroplasts derived from photosynthetic bacteria.
The eukaryotic cell had to develop organelles that included a nucleus plus the mitochondria and chloroplasts in some cells. When the nucleus developed, it's thought that the cell grew larger in size, and eventually got too big. Well, remember in BioH we learned that normally when a cell gets too big, it divides in half. Well, at this time, instead of dividing in half through mitosis, the outer membrane started to infold. This is so that it can still maintain a proper cell surface area to volume ratio. If it has too much volume for the surface area, the cell is considered to be inefficient. So this time we have the outer membrane enfolding, and ultimately this led to the endomembrane system. Remember, the endomembrane system included the nucleus, the rough ER, and the smooth ER, because they all kind of go together. So this concept would explain how all of those structures may have potentially formed. In prokaryotes, Although they lack a nucleus, they do have a general region where their DNA typically sits, and that general region is called the nucleoid region. So if these membranes ended up fusing together around that nucleoid region, we would have a nucleus, and the DNA would be inside. Here shows you what endocytosis is. So this is a review from BioH as well. In endocytosis, a larger cell will push its cytoplasm and its cell membrane out, eventually forming a vesicle around some molecule that's from the outer environment that it's trying to take in. And therefore, it will be brought in as inside of a vesicle. Remember that we learned about this in BioH, and so this is all phospholipid bilayer. This is actually important to the endosymbiotic theory because it leads to the evidence of double membrane bound organelles such as chloroplasts and mitochondria. Remember that those types of organelles actually have two membranes, an inner membrane and an outer membrane. So this is going to help support that theory. The outer membrane likely came from the bigger bacteria that engulfed the smaller ones, so archaea. The inner membrane here likely came from the original smaller bacteria that was engulfed. So here's a couple of important definitions. An endosymbiont, which we've talked about, is a cell that lives inside of another cell because it provides a benefit for that cell. So this relationship, of course, would be mutualistic. They're both benefiting from each other. This image is showing the formation of the membranes first, ultimately leading to endosymbiosis, which is the incorporation of the smaller organisms that ultimately developed into organelles, which would lead us to our first unicellular ancestral eukaryote, which could potentially give rise to future eukaryotes over time. And then this down here again just describes what was occurring. A small aerobic bacteria, which could potentially use oxygen to make a lot of ATP, was engulfed by the larger anaerobic organism, probably the archaea organism. Instead of killing it, it lets it live. It lets it live because it provides that benefit. It's an endosymbiont. Then it gives it lots of ATP, allowing the benefit of the host to be more successful compared to other anaerobic prokaryotes. Eventually, the host cell grows and divides, and as it's doing this, the smaller bacteria inside of it also does that. It grows and divides inside the larger cell. Therefore, it would make more. So you can see like more copies are made essentially of the smaller bacteria. And therefore, future generations would have more and more and more of these particular beneficial endosymbionts. Over time, it did become the mitochondria. And the chloroplast would be similar again, except for the benefit would be different. This time, the smaller cell would be really good, using the sun's energy to be able to make sugars, and then it would give some of those sugars to the larger host cell. There's a lot of evidence that does support the endosymbiotic theory, and these points are listed here. Number one, the mitochondria and chloroplasts that we have today actually do have their own DNA, and this DNA is very different from the DNA inside the nucleus of the eukaryotic cell. In fact, this DNA is much more like prokaryotic DNA. It's circular, like prokaryotic DNA, and it is naked, like prokaryotic DNA. Remember in BioH, we talked about the fact that eukaryotic DNA is not naked. It's associated with histone proteins. And prokaryotic DNA is not associated with histone proteins, therefore it would be considered to be naked. So they do have similar DNA to prokaryotic cells. They also have ribosomes, their own ribosomes that are similar in size to prokaryotic ribosomes. Prokaryotes have small ribosomes, 70S, that's their size, whereas in the cytoplasm of a eukaryote, their ribosomes are larger, 80S. Again, they have a double membrane, an inner and an outer. The outer membrane is similar to the larger archaea organism. The inner membrane is similar to the smaller bacteria that it was originally part of, the one that was engulfed. Since they do have their own DNA and their own ribosomes, they actually can transcribe and translate some of their own proteins using the materials inside of them, which further supports that they likely used to be their own individual organism when they once were bacteria. And then they can only be made if they copy their DNA, divide in half to make more. So therefore, mitochondria and chloroplasts divide or regenerate in ways that are very similar to a bacterial cell would. It's not like the other organelles, which are made by your cells. Your cells synthesize the phospholipids to make more smooth and rough ER today. They make their own lysosomes, for example, but they cannot make more mitochondria or chloroplasts. These must be made by the division of themselves. So that gets us to the first unicellular eukaryotic cell, but at some point they had to become more complex. So these unicellular eukaryotic cells must have been able to develop the ability to reproduce through sexual reproduction rather than just asexual reproduction. And these sexually reproducing eukaryotic cells would serve as the common ancestor to future sexually reproducing eukaryotes. There are current examples of unicellular eukaryotes that can reproduce sexually. In BioH, we only talked about multicellular organisms being able to do this. So remember, we said in multicellular organisms, they would be able to produce male and female gametes, so sperm and egg, which would then come together to create the first cell, fertilized cell, the zygote. But it is quite a bit different in unicellular organisms that can reproduce sexually. In IB Year 2, you will be taught more specifically about this process. Ms. Lockwood will use an example of a single-celled fungus, specifically yeast, that is able to reproduce sexually, even though it's only made up of one cell. At some point, multicellularity had to evolve, right? We don't just have unicellular organisms, now we have multicellular organisms. And this would have had to have evolved repeatedly through many independent events. So it's not like there's a single common ancestor that was multicellular. This happened multiple times throughout evolutionary history. Multicellular organisms are present throughout all 4 eukaryotic kingdoms. So many fungi, many protists, so including some algae, and then all plants and all animals are multicellular. Multicellularity does have advantages. Some of these advantages are listed below. They have a longer lifespan because when one cell dies, it doesn't lead to death of the organism. Whereas in a unicellular organism, it's only made up of one cell, so when that cell dies, the whole organism dies. So this lets eukaryotes live longer. Number 2, eukaryotes that are multicellular have a larger body size. This allows them to exploit niches that unicellular organisms cannot. So remember, a niche is like a little area or space, a micro environment, for example, that is occupied by a species. It's more than just their habitat. It's the resources that they use, the ecological role that they play within their particular ecosystem. And so these types of niches would be different for multicellular and unicellular organisms. Multicellular organisms also have what's called cell specialization or differentiation. This means that they have specialized cells that do specialized functions for them, which leads to complexity in multicellular organisms that wouldn't be present in unicellular organisms. Even though multicellularity does have so many advantages, most of the biomass on Earth still consists of unicellular organisms, which suggests that they must have a relative advantage in some situations over multicellular organisms. And that is it for the notes on the origin of cells.